Hi again. Today we're going to learn about genetic variation, and in particular we're going to learn the mutation as source of genetic variation, differentiate among the different types of mutations, and also learn some of the cool examples related to mutations. This is taking us back to Darwin and this idea of descendant with modification. And descendant, you have to think about you descend from your parents. So we're talking about one generation to another generation. And modification is that we descend from our parents, our biological parents, but we don't look exactly the same. We look different. This is due to the variation that exists. And this is the modification, descendant with modification. So do different species have different genetic information? Well, yeah, right? And individuals have different genetic information? Yeah. So you have different genetic information than your parents or your siblings. Now, so we can understand this a little bit more in detail if we look at this, the balance between complete fidelity of DNA replication and repair, that means the descendant, and a low mutation rate, that's the part of the modification, over long periods of time, allow evolution. Now we put it all together, descendant with modification, of the rich diversity of the species on Earth. In other words, or in a simpler way, all of this diversity that you could see, and if you are in your garden or at home, just look around and look at the diversity that you can see, the biodiversity. All of those species that you have around you or in your street are descendant with modification. And this modification over a lot of time have led to 1 million species, which have been formally described. But there might be more. And now we classified all of this diversity based on how closely related they are. And we have found that there are 3 domains: bacteria, archaea, and eukarya. And we're going to learn more about them in this class. But then, so the modification is the source of all genetic variation. And these modifications, we call them now mutations. So mutations are changes in the genetic code that are going to be responsible for new genes. And which is very important is that if the mutations falls in the gametes, if the gamete has this mutation or carries this mutation, it will pass to the next generation. We have learned that we have sexual cells or gametes and also somatic cells. If mutations happen in the somatic cells, that's not going to happen. That's not going to go to the next generation, that mutation, right? So new mutations, important mutations, advantageous or deleterious mutations are very important if that happens in the gametes. Now, mutations can be of a small scale, and those are called point mutations, and they can change a single nucleotide base pair or more. And also we're going to have larger scale mutations, and those are going to be rearranges of pieces of chromosomes. And we're going to learn about one particular example that involves human evolution. Now we're going to have, in the point mutations, we're going to have substitutions and insertions and deletions. And in the substitutions, we're going to have three types of substitutions. Some that are silent, others missense, and nonsense. Let's see what that means. Substitutions, we are going to have a silent mutation when there is no change in the phenotype. We have the same amino acid. Missense mutation is going to have a change in the genetic information and in the phenotype. In nonsense mutation, we're going to have the change in genotype that causes a stop codon. A silent mutation is going to change in genotype, but doesn't change in phenotype. And that is because if you remember, when we learn about the genetic code, this is redundant. That means that the same amino acid can be coded by different triplets or different codons. So, for example, if we have here a GGC and it mutates to GGA, then the messenger RNA is gonna go from CCG, and it's gonna be the mutation is gonna result in CCU. So we're gonna have proline before CCG, and the result is another proline, CCU. So we didn't have a change in this amino acid. Therefore, it's a silent mutation, right? Nothing changes in this protein or in this polypeptide. We have the same amino acid in the same position. So now let's take a look at this. In a silent mutation, we are going to have the same amino acid. The result of the mutation is going to be the same amino acid. Let's take this for example. We have our double-stranded DNA, right? Then the upper strand is going to function as the template for the messenger RNA, and the result is going to be this molecule that, remember, will have the uracil. And if we look at these triplets, we're going to have a methionine in the first codon, and then AAG codifies for a lysine, and UU for phenylalanine, and GGC for glycine. Now, if we mutate this CCG, because the mutations are going to happen at the DNA level, at CCG, and we have a mutation that instead of a G, the polymerase adds an A, and now we have a codon that reads GGU. And as we saw before, that results in the same amino acid, which is glycine. So there is no change in the phenotype, right? We have a change in the genotype, but not a change in the phenotype. It's the same. Therefore, it's called silent. Now, missense mutation is when we actually have a change in the amino acid. And this is exactly the same example, but instead of the mutation changing G by an A in the third position of this codon, now we have a change of a C. We're going to change a C to a T. So we have T instead of a C. And that results in a messenger that reads AGC instead of GGC. And that change, now it's going to change, or it's going to have an effect on the amino acid. Instead of a glycine, we have now a serine. That is called missense mutation. It changes the amino acid. Missense mutations are very important, even though it's just one simple nucleotide that gets mutated. In some cases, it can have a tremendous effect in the phenotype. this is one example, and a very important example, that is the sickle cell anemia. The wild type, the codon will be, the triplet will be CTT, but then in the mutant we'll have the CAT, and this will be the messenger RNA. This GAA will code for a glutamic acid. But in sickle cell anemia, we have this mutation that changes the glutamine for a valine. And that has a tremendous effect on the blood cells. So normal red cells or blood cells are circular. In sickle cell anemia mutants, we're going to have these sickle red blood cells that they look like bananas to me. So these have also some issues. It represents some health problems to the carriers of this mutation. Now, this was one example of missense mutation, a mutation that changes the amino acid. Not sense mutation is gonna cause a stop codon. Remember, there are 3 stop codons in the genetic code. And then, for example, in the same example, we have TTC here, and in the first nucleotide, we change this T by an A. Now the messenger is gonna read UAG instead of AAG. And this UAG is the signal to the ribosome to stop. So then the ribosome is going to stop and nothing else is going to be translated. Mm-hmm. So as you can imagine, if this molecule, this polypeptide is very important, if we truncate the synthesis, it can have a very deleterious effect for the individuals carrying such mutations. Another type of mutations are insertions and deletions. These type of mutations can lead to frame shift mutations. And some of them can be very disastrous. Let's look at some of these examples. So remember, the red dog ate the bug. We can have a stop codon caused by an insertion. In the past, we have an example of a stop codon caused by a point mutation. This insertion is different because now we have an extra A at this position. And because the ribosome is reading in triplets, right? It just starts with this information, AUG, the methionine, the start codon, and then the next one that is going to be reading is this UAA. This UAA is one of the stop codons. So again, the ribosome is going to stop and nothing else is going to get translated and synthesized. So it's probably going to be a deleterious mutation. But we can also have missense caused by a deletion, for example. In this case, here we have AAAC, and now we have AAC. So we are missing this A. Now, instead of having a phenylalanine, we are going to have a leucine. And then the next one is not going to be a glycine, but an alanine. So we have shifted the frame shift, or we have shifted the read, and now we have something that is very different. Sometimes these mutations can actually have an advantageous, can be advantageous, but often they can be deleterious or have a negative effect. Now, this is a question for you to test your understanding. Which of the following deletions won't change the frame shift? Okay, so the one that won't change the frame shift is D, because then we have here the methionine as before, and then we are deleting these 3 nucleotides. So it's not changing the frame shift. We are going to just exclude lysine, but we're going to have methionine, and then we're going to skip this. We're going to have phenylalanine later, and then glycine right here. Okay. So this mutation won't shift the frame shift. In any of these cases, we'll have a different outcome, and that all of these will change it. So this is how it will look. We'll have methionine, phenylalanine, and glycine. I told you about the small scale mutation, and now we're going to look into the larger scale mutations. And these have an effect at the chromosomal level. And we think that the size of chromosomes or the size of genomes have increased over time due to these rearrangements of the chromosomes. In this case, we're going to have, I'm going to move this over here, we're going to have deletions. So a big portion of the chromosome can be deleted, and now instead of having a large chromosome, we have a shorter chromosome. And this information, all of the genes here get lost. We also can have duplications, which means that the information that we have here, we are going to have it twice. And this results in a longer chromosome, a longer arm of this chromosome. Inversions can also result in inversions are also an example of large scale mutations. And in this case, the information in here, let's say that we have genes A, B, C, D, E. Now we're going to have the order backwards. So that will be D, C, B, A. Insertions can also are examples of large scale mutations. In this case, for example, we can have an insertion of a piece of a chromosome, let's say chromosome 4, into another chromosome, this example, chromosome 20. Now, this is an insertion from other chromosome. We can have also translocations, which that means that one part of this chromosome get into this other chromosome, but the difference with the insertion is that this part of this chromosome gets into this chromosome 4. So the part of chromosome 20 get into chromosome 4. Now we have two chimeric chromosomes. Down syndrome is an example of large-scale mutation. This happens during the migration or the segregation of the chromosomes in meiosis. And it could happen in meiosis 1, like for example here. These chromosomes are not migrating properly. So there is a non-disjunction of these chromosomes, the homolog chromosomes. And also it can happen in meiosis too, in which the chromatids don't segregate to the chromatic poles or to these poles in the cell. And then as a result, we're gonna have an excess of chromosomes in one cell, and other cell or other gamete is gonna have less chromosomes. So if we have 4 chromosomes, we will expect to have 2 chromosomes in our gametes, right? Because we have half of the sets. In this case, we have chromosomes that have, we have gametes that have 3 chromosomes. If that happens in chromosome 21, then we will have a trisomy. This trisomy or trisomic syndrome, it's also called an aneuploidy. So aneuploidy means an abnormal chromosome number. And this trisomy of the chromosome 21 is the responsible for the Down syndrome. We could also have duplications of the entire chromosomes, not only part of the chromosomes as I have explained it, but the whole chromosome sets can be duplicated. And that also or usually happens in polyploids. So polyploids species are species that have an extra set of chromosomes. And we have said that in haploid condition, we'll have 1 set. In diploid condition, we'll have 2 sets. So we are diploid. Humans are diploid. But there are other species that can accumulate more sets. And if they have 3 sets, it will be triploid, 4 tetraploid, and they can keep growing and growing and growing. So plants, for example, a lot of plants can be polyploid, and they can have they can be tetraploid, for example, or hexaploid, that means 6 sets, etc. Now, one example of rearrangement of chromosome structures, it has been hypothesized to happen in humans. Humans have 23 pairs of chromosomes. So we have 23 chromosomes in haploid conditions in our gametes. And chimpanzees have 24, right? So 2 ancestral chromosomes got fused in the human line. That's the idea. So let's take a look to this human chromosome number 2. And just think that the number of the chromosomes are assigned randomly in humans and randomly in chimpanzees. So our chromosome number 2 has this length, and the chromosomes have centromeres. Centromeres are going to have repetitive sequences, and these centromeres are very important because during meiosis in telophase, when the chromosomes are migrating to the ends of the cells, the microtubules, these molecules, these proteins that help with the migration of the chromosomes, get attached into this area of the chromosome, the waist of the chromosome. Now, the other important thing that you need to know is that the telomere sequence are caps in the chromosomes, and they help with aging or preventing aging of the chromosome. And they have this also repetitive sequence. So the information in the telomeres are very distinctive. Which is very important is that in humans, we have this centromere-like sequence in this arm of chromosome 2. And also we have telomere-like sequence below the centromere. This is very interesting because this telomere-like sequence, it shouldn't be the right telomere sequences are in the edges of the chromosomes. So one hypothesis is that the chromosomes 12 and 13 fused, and they fused because they share these sequences of the telomeres, and they fused right here at the edges, and this centromere degenerated into this centromere-like sequence. So if you look at the length, the length of chromosome number 2 matches what it will be the result of adding chromosomes 12 and 13 of chimpanzees. Also, now that we have genomic information of these chromosomes, we can see that the genes and the positions between these two species corresponds. So we have very good case that the two chromosomes in chimpanzees fused and formed the chromosome number 2. So that's a rearrangement of the chromosome structure. So I hope that you have enjoyed this class as well, and I'm looking forward for the next one.